The promise of anti-aging science often begins in the laboratory, where mice live longer, cells rejuvenate, and biomarkers of aging seem to reverse with remarkable consistency. These preclinical breakthroughs generate headlines and hope, suggesting that longer, healthier human lives may be within reach. But moving from animal models to actual human application is a far more complex and uncertain journey, one that faces formidable scientific, regulatory, and ethical challenges. Translating findings from lab to life is not just a matter of scaling up doses or timeframes. It's a profound test of how well biology, technology, and real-world complexity can align. One of the first and most persistent obstacles is the difference between species. Mice are convenient models for aging studies because they age quickly, are genetically tractable, and can be housed and studied efficiently. But they are not miniature humans. Mice live only about 2 to 3 years, and their physiology, while sharing many features with ours, responds differently to interventions. A compound that extends murine lifespan by 20% may show no significant benefit, or even adverse effects, in humans. This gap is not a failure of science, but a reminder that aging is not a single process. It is a multi-system, deeply individual phenomenon influenced by genetics, environment, and time. This is why clinical trials are essential, but they come with their own set of challenges. Anti-aging research doesn't fit neatly into the traditional model of drug development. Most medications are tested to treat a specific disease over a finite period of time. Aging, by contrast, is not classified as a disease, and its progression spans decades. Designing trials to test whether a treatment slows aging, rather than simply treating symptoms, requires long-term studies, large populations, and novel biomarkers. These studies are expensive, logistically complex, and often unattractive to investors seeking quicker returns. Moreover, the current system struggles with the diversity necessary to draw meaningful conclusions. Many early human trials are conducted on relatively healthy, affluent, and often male participants. This limited demographic cannot reflect the full spectrum of aging across different ethnicities, socioeconomic backgrounds, genders, and pre-existing conditions. Without broad inclusivity, results may not generalize to the populations most at risk of age-related diseases, or those who could benefit the most from intervention. Expanding diversity in clinical trials isn't just a matter of fairness, it's critical for scientific validity. Funding also remains a bottleneck. Aging research has historically received less support than studies focused on specific diseases like cancer, Alzheimer's, or diabetes. While interest is growing, much of the current anti-aging work is privately funded by venture capital or biotech firms. This can speed innovation, but it also introduces market pressures and exclusivity concerns. Public investment, through national health agencies and international collaborations, will be essential to ensure that anti-aging science develops with broad accessibility and accountability. There is also the matter of measurement. How do we know if a therapy is truly slowing aging? Lifespan is an imperfect and distant endpoint. Instead, researchers rely on intermediate markers such as epigenetic age, inflammation levels, telomere length, or mitochondrial function, but even these are under debate. Some may be more reflective of health span than lifespan, or they may shift due to unrelated factors like stress, diet, or illness. Without validated, standardized biomarkers of aging, trials risk ambiguity in their results, slowing down the approval and adoption of new treatments. Despite these hurdles, progress continues. Regulatory agencies like the FDA are beginning to explore frameworks for recognizing aging as a modifiable target. Initiatives like the TAME, targeting aging with metformin, trials seek to repurpose existing drugs to delay age-related decline, paving the way for future longevity-focused trials. Meanwhile, the development of artificial intelligence, digital health monitoring, and personalized medicine may help researchers track subtle changes in biological age over time, improving trial efficiency and individual response prediction. What's clear is that the path from lab discovery to human benefit is neither short nor straightforward. But each barrier also presents an opportunity for smarter trial design, better biomarkers, more inclusive studies, and greater collaboration between scientists, clinicians, and patients. If we can align these elements, we may not just confirm the promise seen in mice, we may surpass it. The ultimate goal is not simply to prove that aging can be slowed, but to do so in a way that is meaningful, equitable, and safe for people in the real world. In that sense, translating anti-aging research into human life isn't just a scientific journey, it's a social one, requiring as much wisdom as innovation.